Okay, students, so today we're going to learn about cloning, the process of producing similar populations of genetically identical individuals. So you can see this picture. We're going to show you how you can clone this baby many times, or clone any individual. I'll talk about how it's possible for a female to give birth to herself, to her own twin, and it's possible. Contrary to the science fiction movies you've probably seen, when you have a clone or you make a clone, the clone is not going to be exactly your same age. Okay, it's not like a mirror image concept. Um, it's still your same DNA, but it is going to be born and take the same amount of time to reach your age. So you're still going to be 14 or 15 years ahead of your clone. Okay, so let's talk about how that works. First, you need a human egg donor. So the concept is that you have two people involved, or actually three people involved. You got to remember this for the quiz. First one is the person that's going to be cloned. You're going to have to get the whole genetic information. So you get it from a body cell because the body cells are complete. They're diploid. They have the chromosomes from the mom and dad, and they have 46 chromosomes. So you get it from any body cell. It doesn't really matter where. Skin cells seem pretty good. So you get the cell, and it has its nucleus. And you could take the 46 chromosomes out, or you could take the whole nucleus out. And second thing you need is you need an egg donor. And you wonder why the egg? Well, you're going to make a child from this. So you need, remember, the egg is much bigger than the sperm. And the egg is a powerful cell that's big. It has a lot of machinery like mitochondria, all the organelles that you need to develop a child with. And so that's why the egg is so important. So you get someone to donate an egg. Then what do? Well, you first have to enucleate it. That means enucleate means remove the nucleus. You have to take the nucleus out. Why do you want to do that? Well, you don't want to clone the lady. You want to clone this guy right here, for example. So you have to get rid of her DNA. All you want is just the cell machinery. You don't want the instructions of her. So you take that out. And then you get that enucleated cell, the donor egg, and you get his cell or his nucleus, and you want to fuse them together. You want to basically get his DNA inside that empty cell with no nucleus. And you're going to add some electric shock, electricity that kind of helps them merge together, become a cell, one cell. Okay, so now you have the egg from the female with no nucleus. Now it has the nucleus of the guy. And then you add a couple chemicals. You can do some more electric shock treatment, and then that'll allow the cell to start multiplying and developing into an embryo. Now what do you do with the embryo? Well, you have to get the embryo and implant it into a surrogate mother. It could be the same female who let you borrow the egg, and then you basically put that into her uterus. And why do you need that? Well, because the female has the perfect environment for helping the child develop from an embryo to a fetus. She has all the nutrients, the perfect environment for growing, and all the hormones to help that embryo develop. So once you do that, you allow the baby to develop soon, and then it'll be born. And when it's born, it's the clone of this person here. Now we talked about how can the female give birth to herself. You can just have one person involved, actually. It would be the female here. Let's say we're cloning her. So you're going to take her DNA or her egg. You take out the DNA of that egg, and because it's only 23 chromosomes, right? You want to get the complete copy or complete set. So you get a body cell, same thing. Get a skin cell, take out the nucleus there, and put it in her own enucleated egg, and put it back in her body. This is all can be done with one person, and then she'll have the the embryo develop, and she'll give birth to her own twin. That's crazy. Let's look at the
Well, they say
process
it
a
would
little
work
more.
like this. Start with an egg from a woman's ovary, remove its genes or DNA, then take a cell, like a skin cell, from a patient and put its DNA into the egg. This is called nuclear transfer. Add a few chemicals and the egg starts to divide just like a fertilized egg. After a few days, it becomes a blastocyst, a ball of about 50 to 200 cells. If you wanted to clone a person, then you'd have to place the blastocyst in a woman's uterus. A similar process was used to create the famous sheep Dolly.
So here's a process of enucleation. This is actually a real footage. So watch how they remove the nucleus. Remember, you have to get a donor egg, an oocyte, also called an egg cell, and you got to remove the nucleus because you don't want to clone that. You don't want to clone the donor, right? So here we go.
Oocyte enucleation is the removal of the oocyte's endogenous chromosomes using a DNA stain to first localize the genetic material and a glass pipette for its removal.
Now here's a video of now you're gonna have the empty egg, which is here, and what you're gonna do is add the new nucleus from a body cell, right? From the person to be cloned. Remember why we choose the body cell, because it has all 46 chromosomes. You want the complete set to go in there because you want it to have 46
Cell
to
transfer
develop.
involves placing a cell from the animal we are cloning next to the oocyte cytoplasm.